Good morning, everyone. My name is Ben Willems, and I'm the library's and SynBio application specialist in the COSIA region. And I'm delighted to welcome you all to the TWIST webinar with the title High Throughput Evaluation of Genome Editing Tools. In this webinar, you will hear from our speaker, Dr. Hyongbum Henry Kim, on his research work around CRISPR and Prime editing. Before I introduce him, just a few housekeeping items. All lines will be muted during the webinar. We will answer all questions at the end of the presentation. If you have a question, please submit it in the Q&A box found at the bottom of the screen. Following the presentation, if you could please take 1 minute to do a brief survey, we would really appreciate your feedback. And before I introduce Dr. Kim, I would like to share some latest information about TWIST. My favorite way to start the presentation is by showing you a particularly exciting screenshot from our website. Just because, like all scientists, we at TWIST love to brag about our publication record, which is as of now standing at over 2,500 publications. Considering that we're still a very young company, we only started commercial activities in 2016, we regard these numbers as quite substantial, and we're all very proud of it. With an increase of over 50% just in 2022, these numbers also visualize the steep trajectory at which we have been growing our footprint in the global scientific community. And it is also testament to the fact that we do something very right. And here's what we're doing right. This is one of the most important slides I'm showing you today, as it shows the core technology of Twist DNA manufacturing, our Twist Silicon platform. As all DNA synthesis methods, like the low throughput solution in 96 well plate format shown on the left, our DNA synthesis is still based on the very well-established 30-old phosphoramidite chemistry. However, in contrast to the other platforms, our engineers were able to place more than 1 million spots of oligonucleotide synthesis on each of these wafers. This degree of miniaturization enables a game-changing scale of DNA synthesis throughput. And we're not just changing the game with respect to throughput. By achieving significant improvements in coupling efficiency, Twist DNA oligos are also characterized by an industry-leading quality in terms of 100% oligo representation, so basically no dropouts, very high uniformity, say that over 90% of oligos in a given pool production batch are represented within a narrow bandwidth of not more than 2.5x from the mean yield, and an industry-leading low error rate of only one mismatch on average per 3,000 nucleotides. Using these oligos as the basic building blocks for all products in our portfolio ensures highest possible quality. And here is an overview over our portfolio of products and services. The oligos fresh from the wafer ship as oligo pools, after a thorough QC, of course, to support high throughput applications such as genome-wide CRISPR screens or massively parallel reporter assays. The work presented by Prof. Kim today was also done with the Twist oligo pools. To add further value, the oligos are also used to assemble our gene fragments and clonal genes. They are being used to build a very complex DNA variant libraries with a complexity of up to 10 billion variants, used, for example, in phage display-based panning assays to discover new therapeutic antibodies. Our biopharma vertical is leveraging on the superior quality of these libraries, offering full-service solutions towards therapeutic development for the pharmaceutical industry. The oligos are being modified to serve as probes for our comprehensive NGS portfolio, and we're also venturing into more exotic new business areas such as DNA-based data storage. For example, we have worked with Netflix to store entire TV shows, episodes in HD in DNA, and replay them by sequencing. This year, Twist Bioscience will be the first company in the world to launch a commercial product for DNA data storage. And although this is not really related to the topic of today, it showcases what interesting things one can do with virtually unlimited amounts of highest quality DNA at hand. And last but not least, we're also offering a high throughput expression service for antibodies to support therapeutic development projects. And now I would like to formally introduce Dr. Kim. Dr. Kim received MD in 2001 and a PhD in 2006 from Yonsei University, Seoul. During his PhD program, he studied tissue engineering. After postdoctoral training at Emory University, Atlanta, Georgia, USA, in the field of stem cell biology, he became an independent researcher in 2010 when he changed his research field to genome editing. His laboratory is interested in genome engineering in several types of cultured cells and in mammals, as well as in the development of CRISPR-Cas-based high-throughput methods for genetic studies. So without further ado, let us welcome Dr. Kim to start his presentation. Thank you for being here today, Prof. Kim. The virtual floor is yours.
Thank you so much, Brian. Thank you for the nice introduction. And thank you, Twister company, for giving me an opportunity to talk about my research in here. Let me share the screen. Okay. And today I will talk about high throughput evaluation gene editing tools. And as you know, yeah, I have been in this field of gene editing since 2010, and I have been developing many tools, and part of them are thanks to the contribution of Twist Bio. I appreciate that. First, I'd like to talk about CRISPR nucleases. I think everybody knows it. CRISPR is composed of Cas9 and CRISPR guide RNA and trace RNA. And these two RNA duplexes found in nature can be formed into a single guide RNA, we call it. So only two components required, Cas9 and single guide RNA. And I also, the improved method of gene editing is base editors. The basic of this base edit, base editor cytidine has this amine residue. If this amine residue is removed, then cytidine can be converted into uridine, which is recognized as a thymidine-ase DNA polymerase. Adenosine has also amine residue. If this is removed, then adenosine becomes inosine, which is recognized by guanosine by DNA polymerase. So by deamination, we can convert C to T or A to G. But how can we achieve deamination in a targeted manner? We can combine this deaminase with Cas9 linkase linked with the linkers. And then Cas9 recognizes target sequence, and this C is converted to T by deaminase. Similarly, W also discovered adenosine base editing, which requires deoxyribonucleic adenosine deaminase linked to the Cas9 protein and guide RNA. Then the Cas9 find the target sequence and make the this sequence as a single strand. Then deoxyribonucleic deaminase convert A to inosine, ultimately becomes G. So A to G conversion can be induced by adenine base editing. More recently, already 4 years ago, prime editing has been developed by David Liu's group again. So instead of the deaminase, prime editors used reverse transcriptase. And guide RNA sequence is a little bit longer than single guide RNA used for CRISPR nuclease and base editors. And this longer sequence bind to here, and cDNA is synthesized by reverse transcriptase. And this cDNA sequence, including the red signal, which represent intended editing, can be integrated into the target sequence by complex equilibration. Anyway, so using this prime editing technology, we can induce any kind of small insertion, deletion, substitutions, and in a highly targeted manner. Then, to use these genome editing tools, we need to evaluate them first, because the activity of genome editing tools are not frequent, not 100%. In most cases, they are like 30 to 40% in case of Cas9 nucleases, but in case of like base editors, they range to like 0 to like 80%. And in case of prime editing, usually the efficiency of prime editing is often lower than that of the Cas9 nuclease and base editors, but not always. But anyway, we need to evaluate these CRISPR nucleases. For example, we can evaluate like a CRISPR nuclease first. For this, we need to make a guide RNA. Then we deliver this guide RNA encoding sequence together with CRISPR nuclease, for example, CPF1 or Cas12a. Then we deliver this guide RNA encoding sequence and CRISPR nuclease encoding sequence into cell by transient transfection. Then we wait 3 days, and in 3 days, we harvest cells and isolate genomic DNA and PCR amplify the target sequence using specific pair of primers. Then those amplicons are subject to deep sequencing or T7 assay to determine the indel frequency at the target sequence, which represent the activity of Cas9 or CPF1 nuclease activities at the target sequence. This conventional method is used also for other genome editing tools like base editors and prime editors. However, this tool, by using this method, we can evaluate only one guided RNA activities by one experiment. So this can be compared to the cartridge system. So if you compare these gene editing tools, these seizures, because sometimes people call it genetic seizures, evaluating this process takes a long time and only one by one experiment. And many of them are discarded because they don't have not enough high activities. So only some of them are selected. So to evaluate the process, to change the evaluation process in a high throughput manner, we change the process a little bit. We first generate the guide RNA and target sequence library. To this library, we deliver Cas9 or CPF1 or base editor and prime editors. Then we PCR amplify the target region using common pair of primers. Because we added the target sequence into here, we can amplify the target sequence using common pair of primers. Then we conduct deep sequencing to evaluate the tens of thousands of guide RNA activities by one experiment. To conduct this experiment, we initially designed oligonucleotides. Nowadays we order oligonucleotides, all of them from the Twist Bio, but initially we used a different company. But anyway, we changed to Twist Bio because Twist Bio has a higher uniform lower error rate and higher uniformity. So anyway, these oligonucleotides contains guide sequence and target sequence, and they are paired, guide sequence and target sequence pairs. So we initially ordered the 12,000 oligonucleotides. From these oligonucleotides, we generate the plasmid library of guide RNA and target sequence. This plasmid library we generate lentivirus, which was transferred into 200 T cell to make a cell library. So in this cell library, each cell contains different guide RNA encoding sequence, and also they contain corresponding target sequence. But nothing will happen to this target sequence until we introduce CPF1 or Cas9 or base editors or prime editors. And depending on this CRISPR nuclease genome editing tool activities, editing will be introduced into this target sequence. In case of CPF1 or Cas9, index will be introduced in the target sequence. So to measure this index frequency, we amplify this target sequence using common pair of primers, and those amplifiers are subject to deep sequencing. And we computationally analyze the data to determine the thousands of guide RNA activities by one experiment. So far, I have talked about the method to evaluate the gene editing tools in a high throughput manner. But still, we need to conduct experiment. So we wanted to do something more than that. So we wanted we wanted to not to conduct any wet experiment. Before that time, I watched TV and I saw AlphaGo, a computational deep learning model that play a Go game. Won a game against human Go champion Lee Sedol in Korea. So after watching this event from TV, I studied about the deep learning. So this is what I obtained from my studying. I found out that as long as we have sufficient data, the performance of these deep learning based models are often much better than conventional machine learning algorithms. So because I knew this characteristic of deep learning, so we tried to collect a large amount of data. But if we don't have sufficient data, the performance deep learning is even poorer than that of the conventional machine learning programs. But if we have sufficient data, we have good performance. So we analyzed the CPF activities at 15,000 target sequences, and using this data set and deep learning neural network, one-dimensional deep learning, including one conventional layer and 3 fully connected layers, we developed a computational model that predict CPF1 activity initially in a sequence-based manner. This is the result that we obtained. X-axis represents CPF activity predicted by our computational model. We call it deep CPF1. And Y-axis is experimentally measured in the frequency, individually measured. So that is the method that I just mentioned about it previously. This is a traditional and conventional method to evaluate CPF activities. We observed a good correlation between two values, and so indicating that our computational model is highly accurate in predicting CPF activities. Maybe someone can say, oh, this is not 1, so it's only 0.87. But in biology, even though you repeat the experiment, or if you conduct 2 replicate studies, the correlation between 2 replicates or repetitions experiments are often ranging only 0.8 or 0.9 frequently. So this is, I think, very high as a performance of computational model, deep learning model. So we are the first group to profile CPF activities in a high throughput manner, and using this method, we use deep learning, as I talked just now, to predict CPF activities in a target sequence dependent manner. Then we also applied the same technology to predict Cas9 activities. So we generated deep SP Cas9. Then we also expanded our approach to base editing. So both adenine and cytosine base editors. Then recently we expanded our approach to prime editing to predict the efficiency of prime editing guide RNAs in human cells. So this is also, I will talk about some of the new results that we obtained. We show that so far, okay. So far we have talked about the Cas9 nuclease, but that is SPCas9. But SPCas9 requires NGG PAM sequence. But NGG, so GG sequence can observe every 16 base pairs. But if you consider the other sequence, the opposite sequence, which is a CCN, then we can find the PAM sequence only every 8 base pairs, every 1 out of the 8 base pairs, which is not frequent. So in many cases, you cannot use SPCas9 for gene editing. Only 1 out of 16 cases or 1 out of the 8 cases you can use it. So to overcome this limitation, scientists in this field have generated many different variants of SPCas9, which includes the VRQR variants and XCas9, SPCas9NG. And using these variants, we evaluate, but it is not easy to choose which variant we can use, because the PAM sequence of these variants are often overlapped. So we using oligonucleotide generated from the twist bio, we generate a library of Cas9 library to evaluate Cas9 nuclease activities, and using that library, we evaluate these Cas9 variants with the different PAM compatibilities. We found that SPCas9NG has a widest PAM compatibilities, but at NGG sequence, SPCas9 wild type SPCas9 shows the highest efficiency. So this is the this is first and second nucleotide PAM sequence, and this is third and fourth nucleotide PAM sequence. And the density represents the activity of the selected Cas9 nuclease variant in here. So using these variants, we were able to cover roughly 51% of PAM sequences. 4 nucleotide PAM sequences, the number of 4 nucleotide PAM possible PAM sequence is 4 by 4 by 4 by 4 by. So it's 256, and roughly half of them were able to be targeted using at least one of these Cas9 variants. But still, the other half is not targetable. And after publication of our data, scientists in this field also generated another more variants, which include SPK9 NRRH, SPK9 NRTH, SPK9 NICH, and SPG, and SPRI, and SP++. So NRRH, TH, CH are all from David Lewis group, and Ben Kleinstable's group, SPG and SPRI, and SPC+. And so using these variants, we were able to expand the targetable sequence up to 84%, which is much more higher than this one. And the widest pan compatibility was observed by SPK9 NICH, not SPK9 NG. So this SPK9 NICH showed the widest PAM compatibilities as compared to other variants. So using this expanded Cas9 variants with different PAM compatibilities, we can expand the range of base editable target sequence. For example, if we use SPK9 based base editors, which has NGG as the PAM sequence, we can target only 9% of C2T conversions for pathogenic or likely pathogenic single nucleotide polymorphisms reported in ClinVar. And also in A to G conversions, only 8% can be targeted using base editors. And C2G conversion, only 7% can be targeted using SPK9 based base editors. However, if we use these Cas9 variants with different PAM compatibilities, now we can target 83% and 84% and 83%. So roughly 83 to 4% of single nucleotide polymorphisms, C2T, A to G, or C2G mutations can be targeted using our base editor. Our base editors are complexed with different PAM compatible Cas9 variants with different PAM compatibilities. And so far we have talked about the high throughput evaluation generating tools. And let me switch gear, and I will talk about a functional evaluation of various variants of unknown significance. So by using the next generation sequencing, actually we should change the term. It's already current generation, so we can say, yeah, current generation sequencing or deep sequencing. Anyway, this new sequencing technology, nowadays we are finding variants in human patients or normal humans in a highly acceleration speed. But we don't know of the function of those variants. And I think this is one of the biggest problem in current genetics. This problem of various variants of unknown significance is especially important in cancer biology. So this is a cancer cell. So single cancer cells contains more than tens of thousands of mutations. However, this single cancer cell, but though the cancer driving mutations are very minority of them, and the other mutations are thought to be all passenger mutations, which accumulated over time from fertilization and childhood, adult, and aging. So But so we wanted to know which mutation is really cancer-causing mutation and which is not. But single cancer cell contains at least tens of or hundreds of mutations. So to evaluate each functional variants or mutations, we need to introduce one variant into one single cell, then observe the phenotype of the cell. This is a standard to determine the function of the variant. And for this, we used base editors. So first, we generate cells that expresses CBE, cytosine base editor, or ABE, adenine base editors, in a doxycycline responsive manner. So this is an RTTA sequence is integrated into the cell. And if you add doxycycline, this RTTA binds to the doxycycline and binds to activate the RTTA binds to here and transcribe this sequence. This is AB CB cytosine base editor, and this is adenine base editor. And cells that do not express these base editors were removed by hygromycin selection. So these cells that we generate express base editors in response to doxycycline. Then we generate a guide RNA library that would induce mutations observed in cancer cells. For this, we generate the guide RNA library using oligos from Swiss Bio. And in addition to guide RNA sequence, we added also target sequence to monitor the activity of this guide RNA sequence. So we to the cells that express base editors in response to doxycycline, we transit this guide RNA library containing the corresponding target sequence. Then we add doxycycline for 10 days to induce base editing. Then this cells contains each of cell contains variants observed in cancer cells. Then these diverse cells were cultured for 2 weeks in the absence of puromycin or doxycycline. Then we harvest cells and analyze those cells to determine which guide RNA are enriched or depleted, which means which variants generated by the guide RNA are enriched or depleted. So for this one, Hyongbum and Sungho in my lab performed this experiment, and x-axis is normalized log fold change, and y-axis represent a p-value. So black dot represents negative control, non-targeting guide RNAs. Most of them are neutral in here. So log fold percentage is almost 0. But some guide RNA shows outflowing phenotype, and some guide RNA show depleting phenotype. And majority of them are neutral in here. For this is for cytosine based editing. This is adenine based editing. So we call it library A, library C. Cytosine based editing, but a similar phenotype. Anyway, non-targeting RNA shows almost 0 log fold change and also low p-value. I'm sorry, high p-value, which is not significant. So using this volcano plot, we classified guide RNAs from depleting, neutral, and outgrowing, and likely neutral, and likely depleting, and likely outgrowing. So we classified into 7 functional groups. But so far, we have classified, using this method, we classified 62,000 guide RNAs. But so far, we have classified only guide RNAs. But single guide RNA can induce several variants. For example, in here, this is the PAM sequence. And if you use adenine base editing editor, ABE, this is a target window. Target window is not like a one base pair. It ranges several base pairs. And this is target window. In this window, we have 3 A's. So 3 A's, all of them can be converted into G, but that is minor case. Only one of them can be converted. The middle one, or the first one, or the last one. So there are 8 different variants can be generated by a single guide RNA. But we don't know, so we need to find, but in this case, this is dominant variant generated by this base editor. In addition, more importantly, base editing efficiencies are not 100%. So this is base editing efficiency that we measured depending on the target sequence. At the target sequence located in position 5 and 6. So PAM sequence is here. So position 20 is next to the PAM sequence. And this is the position 1 is the starting point of guide RNA binding sequence. So position 5 and 6, base editing frequency is pretty high, like 70 to 80%. But at some base editors, still, even in the peak of the base editing window, some base editors show the low frequency, like 20% or even lower than that, both for library A for adenine base editing, or library C for cytosine base editing. So base editing efficiency are sometimes not enough. So far we have classified the function of sgRNAs, the phenotype. But what we want is the function of the variant. Sometimes if sgRNA base editing induced one variant by one guide RNA, the variant and phenotype can be linked. However, if sgRNA cannot induce high base editing, then variant cannot be linked to the phenotype. So if we link, then this becomes false negative. So cannot be linked. And sometimes sgRNA can induce several variants without any dominant variant. In that case, the phenotype of this sgRNA can be explained by some of the function of these variants. So this case is hard to interpret. So we discarded these guide RNAs, and we also discarded the inefficient guide RNA. Then we chose only these variants. So out of 62,000 guide RNAs, we chosen 35 sgRNAs, which induced one major variant, only one major variant. So we were able to classify 35 variants by high throughput manner. And we found that majority of them neutral or likely neutral phenotype. And very minor fraction, which is 0.1% of variants observed in cancer cells, can induce outgrowing phenotype. And so some mutations found even in the cancer cell cause depleting mutation, which is which can have anti-cancer effect anyway. So let me and this is our lab members. I'd like to thank all of my lab members, especially Yonggang, Seungho, and Nahe, whose result just I talked about just now. And also I have many collaborators. I thank all of them. And thank you for your attention, and I will be happy to take any questions.
Well, thank you so much, Prof. Kim. That was a very impressive presentation of your even more impressive work. Thanks so much. So we're now going to dive into some of the questions that have come in. And please remember, you can add your questions in the Q&A box at the bottom of the screen, and we will try to get through as many as possible. And I'll give the audience some time now to enter their question. And at the same time, I'll start reading.
Yeah.
Or would you like to make a remark, Prof. Kim?
Yeah. First question is about the technological question. It's about how to prevent recombinations during PCR. We minimize the PCR cycles when we perform PCR to minimize recombinations. And that is the first one thing. Yeah. That is what the technology we are using. So simple. Yeah.
Okay. Thank you so much. As you already answered the first question, and for the audience, let me read the question again. So it was so one question regarding the high throughput libraries. How do you prevent analyzing recombinations during PCR leading to false positives? And that has been answered by Prof. Kim already.
Yeah.
We have another question, which is, are there efforts to find more variants of SPCas9 to target more PAM sites?
Yeah, that is a great question. I think some scientists may work on that topic. Yeah, so we don't know the future. Yeah, but I believe some people are still working on that topic. Yeah.
A related question to this is, now that we see that different variants of Cas9 and PF1 are targeting different PAM sites, would it make sense to co-express several Cas9 variants together in each cell to increase the efficiency of targeting a bigger variation or a bigger share of PAM sites?
Yeah, that's an interesting question. That's an interesting question. I think we never tried that one, but maybe we can do that. That is a great idea, I think. So far nobody did that work.
Okay.
Yeah.
Well, maybe we'll connect you to the person asked this question, and you can maybe write a paper together after a fruitful collaboration.
Yeah.
There is another question, and it is also about combination. So you showed your experiments in which you look for the different mutations in a cancer cell to decipher between passenger mutations and cancer-driving mutations. Now, it is often said that it is the accumulation and the combination of many mutations together that start the proliferation, the wild proliferation, or that start the cancer. Now, here again, with the dual guide or even triple guide application in the toolbox of base editing and prime editing, would you also think about running more complex experiments where you use dual guides or triple guides to target more of these potential passenger mutations to see if synergistic effects of these mutations also drive the cancer emergence?
That is great question. One of my colleagues also raised a similar idea of targeting using CRISPR to target those cancer specific passenger mutation. I think that is interesting study. I'm sorry. Interesting study to conduct. We haven't tried it yet so far, but in the future, maybe, we or other people would be interested in those studies.
Okay, great. I think the community is looking forward to seeing more publications in this field. All right, I have one more question, which is: are these guide RNA target libraries available in Edgy?
I see. We tried, but the plasmids are available, but libraries, it is difficult to maintain the like a uniform homogeneous distribution of the each component of library. So that can be available from our lab if you are interested, but not from the Edgene.
Yeah, I think everyone who's interested in getting all that sequence data could reach out to you, and you'll send.
Sequence data are available, all available. Yeah.
Okay, and they're probably also available in the supplemental materials of your publications.
Yeah.
Okay, great. We have a couple more questions that have come in. So the next one is: which step in the process of performing library screens is the biggest bottleneck or challenge when planning to go for larger libraries? Is it cost of the pools, the cell culture effort, or the sequencing costs?
That is a great question. Cost of pool also definitely important. Also sequencing costs a lot, and also we have to culture. All of them cost, cell culture and sequencing and the oligonucleotides. All of them, each component cost a lot because we perform in high throughput in a manner. So, and also designing is also very important. You want to design a good library. The designing also important. So maybe biggest bottleneck is, I think, maybe, but all of them are bottlenecks. So designing, culture, and sequencing, and oligonucleotides. So you cannot choose only one.
Okay, great. Thank you for answering this question. Here is another one. So the anonymous attendee writes: "Hi, Prof. Kim, great talk. I didn't have a specific question, but more generally, do you feel the rates of off-target effects are a serious concern with CRISPR technology?"
That is a great question. Actually, I don't think off-target effects is the most important or serious concern in this field of CRISPR technology, especially you are interested in applying this technology in gene editing therapy in human patients. I think still, yeah, off-target effects are still important, but in more cases, I think efficacy or editing efficiency, which affects the therapeutic effects, are more important because in many cases that we can predict the off-target effects pretty well, and we know that the method to design to avoid off-target effects. So I think off-target effects would not be the biggest concern in the field, but it's still important issue. Yeah, still important, I think. Yeah.
Definitely. Also very, very important when considering to bring CRISPR-based gene editing into a clinical application for cell therapy. Okay, we have a last question. I'll read it. First of all, nice work. What are the advantages using base editors other than Cas9-KO approaches when evaluating gene mutations?
So, yeah, that is a great question. If you use Cas9 knockout approaches, you can determine function of the dead gene using knockout approaches. But if you use base editor, you can determine not only the function of gene, you can determine the function of the variant of the gene that can be generated by base editors. So even narrower range of functional evaluation can be achieved by base editors.
Okay, thank you very much. And we've just got one more question that came in last minute. I'll read it to you. So in the last slide, you show that very low percentage of the guide RNA and its variants resulted in depletion or outgrowth. This result is interesting. Have you looked into where these variants are located? Perhaps at the regulatory elements of oncogenes? Have you looked into the biology of how these variants work on cells?
Yeah, that is also an interesting question and great question. And yeah, when we not the depletion, it's the outgrowing low percentage of guide RNA induced outgrowing phenotype was associated with like a cancer-related gene. For example, P53. To my memory, the P53 is the most frequent. The variant of P53 was the most frequently observed, and other cancer-related genes were also observed as outgrowing phenotype. So, but all of them, P53 dominates as a cancer-relevant gene. Yeah. By our data.
Mm-hmm. Okay, great. Thank you for answering this question. And as it looks here, this was the last question. So this brings us to the end of our webinar, and I'd like to thank all of you for joining us today. We hope that you have enjoyed this Twist webinar and would really appreciate your time completing a short feedback form about the event. Your feedback is important to us so that we can improve future Twist Bioscience events. Please click the feedback link that will be appearing on your screen to access the form. If you would like more information about how Twist products can help you in your research, please visit www.twistbioscience.com. So thank you again and enjoy the rest of your day. And of course, once again, I would like to thank Prof. Kim for giving that very nice talk. Thank you very much. And have a good day.